you can let me know if you can. Okay, if you can see Nikki's message in the meeting chat where she has pasted the Google Docs link for the attendance, and if you could just put your name and affiliation on that attendance list, that would be great. All right. So I'll give everybody a minute or so to do this. So you can just put it in the whole block under the list of names and attendees. Yeah.
I'm just going to stop screen sharing to adjust my monitor, but I'll put it back in 1 second.
Okay, no worries. Thank you.
It's just running off the edge of my screen, so I can't see my slides very well. But now I fixed it. Let me re-screen share. Okay. Also, the power cord wasn't working, but this wasn't plugged in properly. I think I saw.
Going to be one of those days, perhaps. Hopefully not.
Yeah. Why is there a giant dinosaur outside? Just kidding.
All right, so I'm just gonna, as everybody's just finishing up typing their names, I'm just gonna get started because we only have an hour and I'm sure we all have a lot of questions. So I'm very excited and honored to introduce our guest speaker today. Professor Ed Boyden completed his undergraduate degrees in physics and electrical engineering and computer sciences and a Master of Engineering at MIT. He completed his PhD studies as a fellow in the neurosciences program at Stanford. And Professor Ed Boyden also joined MIT as an assistant professor in 2007 and is now a professor in the departments of brain and cognitive sciences, media arts and sciences, and biological engineering, and an HHMI investigator. He has made great strides in developing tools to allow researchers to better study the brain. Of note is expansion microscopy, of course. So please join me in giving a warm welcome to Dr. Ed Boyden for his talk about expansion microscopy, development and application.
Great. Yeah, thank you so much for the invitation to come and speak. And yeah, happy to get to talk to you today. And hope there are lots of interesting questions. And feel free to, I guess, enter them in the chat window. That's helpful, or we'll take questions at the end because it can be hard to call on people over Zoom. So our group at MIT works on technologies for imaging and controlling biological systems. And so we've built a couple tool suites now that are in widespread use in neuroscience and biology. One of our earlier projects, optogenetics, allows us to control the brain cell electrical activity with light, and that's being used by thousands of scientists now. More recently, we've been focusing on imaging of biological systems. And so today I'll focus on one of those tool sets, expansion microscopy, that's also been spreading very rapidly throughout biology and both basic and applied. So basic idea is, in contrast to the last, really several hundred years of microscopy, where, imaging relies on magnifying an image with a lens, we're trying to magnify objects directly. And the reason we started this project was because of the difficulty of nanoimaging. So biological molecules, genes, gene products, other biomolecules are very small, nanoscale, and they're often organized with nanoscale precision. But if you want to image them in a cell or a tissue or an organ or even an entire organism, that's difficult. And so we set out to try to develop a real strategy that would allow anybody to do nanoimaging. And hopefully to democratize nanoimaging. And the basic concept is we take a specimen and we synthesize a swallowable polymer throughout it. So this is a polymer that first order is kind of like the stuff in baby diapers. It's a swallowable material, and we do it just right. We weave chemically this polymer inside cells and outside cells, around biomolecules, in between biomolecules, very, very densely. The spacing between polymer threads is only 1 to 2 nanometers on average. We anchor key biomolecules to the polymer. We soften the specimen through enzymatic or heat or detergent treatment, and we add water to expand the specimen. And that allows an even expansion of a biological specimen down to nanoscale precision. So how does this really work? And what are the steps really like? Well, like all technologies, it owes a debt to earlier approaches. In 1980, a physicist at MIT named Toyoichi Tanaka was working out the phase transition-like physics of ionic gels. These are hydrogels with highly charged polymer backbones. And so this cartoon illustrates the kind of thing that he studied. You could take sodium polyacrylate, a highly charged polymer, the active ingredient in baby diapers, add water, and the water is drawn in through osmosis. The polymer threads are highly charged, and they separate apart from each other. So this gives this enormous expansion within a matter of minutes. And furthermore, the physics of this is well behaved, and that was one of the things that got us thinking, well, maybe this is something that we could try to apply to evenly expand a biological system. Now, you can't just dump the baby diaper polymer on top of the specimen. You have to get it inside the specimen, inside the cell, in between the biomolecules even. Well, interestingly, in 1981, a paper from Christine Dreyer and Peter Hausen showed how to do just that. They took preserved amphibian embryos, tadpoles, and synthesized a polyacrylamide hydrogel throughout them. They then went on to show that this could facilitate staining and imaging. So they used polyacrylamide. We're using the charged cousin, polyacrylate. But it's fun to think that if somebody put 2 and 2 together, the talk that I'm giving you today could have been given over 40 years ago. So in a nutshell, what we want to do is to take a cell like the one on the left of this cartoon and pull apart the building blocks of life so they look like something on the right. A constellation of biomolecules hovering in space. Their relative organization is preserved, but they've all been pulled apart from each other. So 2 biomolecules that we're touching are now some minimum distance apart, and 2 biomolecules that are some distance apart have been scaled up by a linear factor. So, of course, this only works on preserved specimens, not living ones, but allows you to get a molecular map of what is inside the cell, tissue, organ, or organism. The following 4 cartoons illustrate how the technology works. And all expansion protocols, our group, of course, developed the first one, but now many groups are developing variants that are customized for different purposes, like expanding bacteria or expanding bone. But all protocols more or less have the same kind of steps. In the first step, you attach some kind of anchoring molecule to biomolecules of interest. So in this cartoon, proteins are shown in brown and anchors are shown in purple. Basically, we're giving a little handle to each biomolecule type. And so now there are anchors developed by our group and others that bind to sugars and lipids and DNA and RNA and proteins, and the list goes on and on. In the next step, you have to form that swallowable hydrogel. We do this using a method called polymerization. So we add monomers, shown as little white spheres, and they self-assemble through a process called polymerization into these polymeric threads, and they weave their way in between and around the biomolecules. When a thread encounters one of those handles or anchors, they form a molecular bond. In step 3, we soften up the specimen. We can use heat, detergent, or enzymes, or all 3, to loosen up or even destroy molecules so we can soften the specimen. We can then add water, and the baby diaper polymer will expand, but because of the softening, because of the anchors, and because of the swallowable hydrogel, the specimen will become larger. The biomolecules will be pulled apart from each other. So we announced this discovery that we could physically expand biological specimens in 2015. This was done by 2 then graduate students in my group, Fei Chen and Paul Tilburg. In panel B is a pretty large piece of the mouse brain, several millimeters on a side. Panel C the same piece of mouse brain tissue that's been expanded by about 100 times in volume, by about 4.5-fold in each direction. The specimen starts out with the polymer in a compact state, shown in the upper left. The spacing between polymer threads is around 1 to 2 nanometers in size, smaller than many biomolecules. And then after we expand, the baby diaper polymer, the sodium polyacrylate, will attain an extended conformation. Here's a video of a small piece, actually a pretty big piece of the mouse brain being expanded. We formed the swallowable polymer earlier, and then we add the water right there. Right there, actually. Okay, I hope you can see that this piece of the brain is swelling. It's a 30-minute video sped up to 30 seconds, but I hope you get the flavor for the rapid changes in the volume that occur. So the first question we got in the early days was, is the expansion even, or does it introduce error? So we spent a lot of time taking pre-expansion images with a classical nanoimaging method. There are classical nanoimaging methods like super resolution microscopes, although they do require equipment, sometimes very expensive equipment, and they can be quite slow, especially if you're trying to image a large 3D object. But you can take a pre-expansion image with a classical super resolution method, expand, and then take a post-expansion image with a regular microscope. So here we're using some cells with proteins, in this case microtubule proteins, decorated with fluorescent antibodies against tubulin. In light microscopy, for the most part, we're imaging fluorophores or other contrast agents that are bound to biomolecules. And we took the pre-expansion image, in this case with a SIM super resolution microscope, shown in purple, and then in green and overlaid in these images is the post-expansion image taken with a regular old confocal microscope. And so the distortion is not zero, but it's very small, just a few percent over a typical microscope field of view. And it turns out for almost every question that we're looking at in biology or in medicine today, this kind of precision is enough. Nevertheless, we are working to improve the polymer, as I'll talk about later. So that's distortion. What about resolution? So resolution is how close two things are until you can no longer tell them apart. We can look at known structures like microtubules, where we have a ground truth knowledge thanks to decades of classical imaging with super resolution microscopes or electron microscopes. We can take our image, expanded, in this case 4.5-fold in each direction, and image on a regular confocal microscope. Oops. And then we can take what we judge the microtubule width to be and divide it by the known ground truth, or deconvolve it more accurately. And we can then estimate how much error or blur has been introduced at the nanoscale. And so we get an error of around 70 nanometers or so. Now, if you think about it, a typical good confocal might have a lateral resolution of 300 nanometers. We're expanding by 4.5. 300 divided by 4.5 is about 70 nanometers. Earlier, I told you that the polymer spacing is 1 to 2 nanometers. So if we expand more, can we get even better resolution? As I'll show you later, the answer is yes. But even with 4.5-fold expansion, there are many things you can do that are difficult and sometimes impossible to do with earlier techniques. So here's a piece of the mouse brain. On the left are unexpanded images. On the right are expanded images. It's a couple millimeters on a side, so a pretty large object. The color code is shown on the left. So we're using fluorescent antibodies to decorate proteins. These are brain specimens. So we're looking at synaptic proteins. Bassoon is in blue. Homer 1A is in magenta. We label some fluorophores with an antibody against the fluorophore to make it brighter. So you can see yellow fluorescent protein as well. Each white square is zoomed in the image below. So the left is unexpanded and the right is expanded. And I hope you can see that as we zoom in top to bottom, the left images get blurry. We're hitting the resolution limit of the confocal microscope, but on the right, the images get crisp all the way to the very end. In fact, the purplish blobs in the left can be resolved as blue and pink subregions on the right. And those are pre- and postsynaptic protein domains. Blue is bassoon, a presynaptic protein. Magenta is Homer 1A, a postsynaptic protein. If we measure the distance between these protein densities, we get exactly the same answer that Xiaowei Zhuang and Catherine Dulac measured many years ago with storm super-resolution microscopy, invented by Xiaowei, but this time we can make the measurements on an ordinary confocal microscope. So one take-home message is that now regular microscopes can do nanoimaging, but also you get a speed up because a classical microscope can go basically as fast as you want, right? If you're doing classical super resolution imaging, you're basically in some forms like Storm or Palm imaging individual molecules. But if you're scanning a laser or scanning an array of lasers or doing other classical diffraction limited methods, you can go at blazingly fast speeds. In fact, one interesting side effect of expansion, if you recall, we're expanding in water, right? It's going up by 100-fold in volume in water. That means it's mostly water. So it becomes transparent. On the left, up in panel A is a small piece of the mouse brain, actually a pretty big piece of the mouse brain. Panel B, we expanded it 100 times in volume, so it's 99% water. It becomes completely transparent. That means you can use other tricks that go at even faster speeds. So there's a trick called light sheet microscopy, where you shine light from one angle, and you take pictures from a 90-degree angle. And so every picture has a full 2D sheet through the specimen. You can go at very fast speeds because of this. And these microscopes are commercially available. We're working with Eric Betzig, another super resolution pioneer. We use one of his light sheet microscopes called a lattice light sheet microscope. And what we found is that if you expanded and then image on that microscope, you could go nearly 1,000 times faster than a classical super resolution technique. So here in panel C, we're decorating mitochondrial and lysosomal proteins with antibodies against them. In the lower part of the slide, we're decorating myelin proteins. This is work done by Ray Gao, Sho Asano, and Goku Upadhyula, working with our group and Eric Betzig's group. And so, with the great improvement in speed, you can also image very large volumes. So here, we're looking at the thickness of the mouse cortex, layer 1 on the right, layer 6 on the left. Again, a greater than 1 millimeter sized object, but we can get down to nanoscale precision. I'll just show you a brief movie clip where we're zoomed in very, very close here. Pink and blue are again the pre and postsynaptic proteins. Yellow is YFP, yellow fluorescent protein filling neurons. We can zoom in and get all the way up to individual synaptic connections. But we can also take a step back and zoom out and see the context of the synapse or the synaptic proteins in the local circuitry. Eric, being at the time at Junelia Research Campus, we worked on the fruit fly brain as well. Here's the Drosophila fruit fly brain with the dopamine neurons expressing a fluorescent protein. Not shown here are also the synapses, which were labeled by an antibody. And we're just going to do a fly through of the fly brain. So we're now zoomed out. Now we're going to zoom in and fly into the middle of the fly brain. Now we're going to go out to the side. I hope you can see that we can resolve axons, dendrites, and so forth quite well. But we can zoom out and see the context of the entire brain as well. So expansion not only democratizes nanoimaging for ordinary microscopes, but if you're willing to use a specialized microscope, you can image very large-scale objects, even entire brains in their intact state in a single setting. So far, everything I showed you is imaging proteins decorated with antibodies or fluorescent proteins. This is what we call protein retention expansion microscopy. It's a very established protocol. I would estimate probably about 1,000 groups are doing it at this point. And it follows the same kinds of pipelines that I explained in the animations earlier. You can anchor, polymerize, soften, and expand. You can apply fluorescent labels either at the beginning of the process or at the end, depending upon what your scientific goal is. You can do it with antibodies that bear fluorophores. You can also do it with fluorescent proteins, some of which will survive the expansion process. You can also decorate those fluorescent proteins with antibodies bearing fluorophores if you want to boost the brightness. So for example, here's an example where we express fluorescent proteins in cells in the living mouse, in this case in the brain. We then decorate those fluorescent proteins with antibodies. We can then expand and image and look at the wiring of the brain. So this is the mouse hippocampus. We express fluorescent proteins with viruses. If you zoom in with a confocal microscope, you quickly hit the resolution limit. But if you expand first and then image, you can cleanly see the wiring of the brain. Now, everything I mentioned so far is looking at proteins, but we can also look at other things like nucleic acids. Oswasi joined the team and worked on expansion microscopy of nucleic acids. And what you can see here is that we have different anchors that bind to different kinds of biomolecule. In fact, now in recent work from our group and others, there are now universal anchors that can bind to many kinds of biomolecule. But you can apply the anchor, it'll bind to DNA, RNA, proteins, sugars, lipids, and beyond. You form the polymer, soften, and expand. And then we can do post-expansion in situ hybridization. That's where you bring in a probe against the nucleic acid of interest, and you can then see it with a fluorescent microscope. So here are many examples of pre versus post-expansion imaging on a confocal microscope with in situ hybridization. And I hope you can see that we have great yield, as shown in the upper right, but all the post-expansion images have better resolution than their corresponding pre-expansion images. Same specimens, same field of view, same microscope. Those are in cell culture. You can also do this in intact brain or other tissue. Here we're anchoring and expanding proteins as well as RNA. So you can see the yellow fluorescent protein in green, and then in purple, we did post-expansion in situ hybridization. In the initial paper in 2016, we use different anchors for the protein and the RNA, but in recent work that's on bioRxiv from Gaoqi Yang and Yi Kui in our group, we have a universal epoxide-based anchor that can bind to many kinds of biomolecule, including proteins, nucleic acids, and beyond. So far, everything I've showed you is expanding by about 4-fold. Can you expand more? Well, you can change the polymer recipe and expand up to 10-fold. Several groups have done that. But if you, and there's protocols like X10 and T-Rex and so forth that are in the literature. But if you want to expand greater, the problem is, the original polymer eventually starts to fall apart. And so in our group, we were wondering, well, can we expand something infinitely? Can you expand something basically as big as you want? So when JB Chang was a postdoc in the group, we had the idea to expand something over and over again. Take a specimen, polymerize, and expand it, like I showed you before, form a second polymer in the space opened up by the first expansion and expand it again. And you can do that a third time and a fourth time and so forth if you want. Now, recently, Deblina Sarkar, Jinyun Kang, and Oswasi in our group, with great help from Margaret Schroeder and others, we showed how to do this with all off-the-shelf chemicals. So I'll talk about their work now. This is recently published in Nature Biomedical Engineering. So we can take a specimen, polymerize and expand it, form a second polymer in the space opened up by the first expansion, and expand it a second time. And this is a version we call expansion revealing, because it illustrates a second benefit of expansion. So expansion democratizes nanoimaging, but it also pulls things apart from each other. We wondered, labels, of course, have to bind their targets in order for light microscopy to work. Maybe by expanding things away from each other, could we get better labeling? The cartoon at the top illustrates how this works. In the top row of the cartoon, the green and red antibodies decorate proteins on the outside of a complex, but they can't get inside. If we pull the proteins apart, though, now the yellow and blue antibodies can penetrate into the complex and label their targets. Now, many proteins, of course, are connected to other proteins. That's how many of them work, right? Biomolecules work by talking to other biomolecules. And by talking, I mean binding and altering each other, right? So we wondered, in neurons and other specimens, there are all sorts of examples where molecules are jam-packed together. Maybe these molecules would benefit by being expanded away from each other and in the decrowded state being labeled. So working with Tom Blanpede at U Maryland and others, we decided to try to figure out if some of these proteins at neural synapses were too crowded to label, and if decrowding them from each other would benefit the specimen labeling and maybe even make invisible proteins into visible ones. This is a crowded slide, but bear with me. Panels B, C, and D show a couple examples of proteins where the pre-expansion staining, shown in yellow, and yes, there's not much yellow, is pretty dim. Post-expansion labeling is shown in pink, and it's much brighter. So panel B is a calcium channel, panel C, RIM1, A, and 2, a presynaptic protein, panel C, PSC95. In all these three cases, we get much better staining with post-expansion staining than with pre-expansion staining. And by the way, the fluorescence goes up at the synapses. We have reference stains that don't change with pre versus post-expansion staining, and we see the increase of the stains at these specific locations. So we don't think it's non-specific staining. As I alluded to, not all proteins are crowded, so panels F, G, and H are examples of proteins where the pre and post-septic stains are much more similar to each other. Homer, bassoon, and Shank 3. Now this is important because many proteins attain their function through nanoscale connections and coordinations with other proteins, right? That's how many proteins signal. We use the fact that we can now see proteins that are hard to see, like calcium channels, through post-expansion staining, and showed that, for the first time to my knowledge, that such proteins are nanoscale coordinated in space with other kinds of proteins. So to my knowledge, this had not been studied in intact brain tissue before our study, but we were able to use post-expansion staining of multiple kinds of proteins that were previously difficult to visualize and show that they have coordinated distributions of proteins. They're not randomly distributed, they're coordinated, which might help with the precision of brain signaling. Now, in brain diseases and other diseases, there are lots of examples of proteins that are aggregated or densely packed. Amyloid plaques in Alzheimer's disease, alpha-synuclein in Parkinson's disease, the list goes on and on. We wondered if we expand those, could we get better labeling? Working with Li Wei Sai at MIT, we looked at Alzheimer's model mice. So these are mice engineered with genes that mimic some of the effects of Alzheimer's disease in human patients. We used a 5X FAD mouse that has amyloid plaques. Pre-expansion staining is shown in yellow. We use an antibody against A-beta, one of the major components of amyloid plaques. The pink images are with post-expansion staining. Same antibody, same specimen, same field of view, same microscope. I hope you can see that the pink images have much more detail than the yellow ones. In fact, if we zoom in, we found even these nanoscale features, these fractal-like dots that were clear with post-expansion staining, but which were very difficult to see with pre-expansion staining. This held for multiple antibodies against A-beta, and we never saw them in wild-type animals. So we're very excited now about the ability to look at all sorts of different pathological states through decrowding to label proteins better. Pablo Valdez-Quevedo, a neurosurgeon who worked with us, wondered, well, if we could see invisible proteins, what about invisible cells? As a neurosurgeon, he works with patients who have brain cancer a lot. Brain cancer cells, the aggressive ones, are characterized by certain dense proteins, GFAP and vimentin, and these are images on the left from actual human patients of brain cancer specimens. We wondered if we expanded the brain cancer biopsies and labeled the vimentin and GFAP with antibodies post-expansion, will we get better labeling? Now, the top three rows, A, B, and C, are pre-expansion labeling of these human cancer biopsies. Rows D and E are the same specimens, same antibodies, same microscope, but with post-expansion labeling. We get much better labeling. We wondered if we're getting better labeling of these aggressive tumor markers, would it increase the tumor cell counts? It's a crowded slide, but on the right-hand side, you can see where my mouse is pointing, the vimentin and GFAP double positive cells. These are the putative aggressive tumor cells. Pablo convened a team of people to count the aggressive tumor cells, either with post-expansion staining or with pre-expansion staining. Amazingly, the number of aggressive tumor cells or putative aggressive tumor cells, because of course we don't know for sure what these cells are, right? Maybe it's a new kind of cell that nobody's ever described. But the number of putative aggressive tumor cells when done with post-expansion staining was 6 times higher than with pre-expansion staining. And by the way, that was after we expanded, stained, and then shrank the sample back down. We wanted to know whether the better staining was helpful even if you didn't care about resolution. So what Pablo did in row D in the lower left-hand corner was he expanded these brain cancer specimens, labeled them, shrank them down by adding salt, and then imaged them. So even if you don't care about the resolution, you might care about whether you're labeling proteins accurately. And I should point out that all these comparisons were done comparing it to classical antigen retrieval. So we don't think this is just antigen retrieval effects. We do think this is decrowding of proteins from each other. So in summary, I told you that we have built expansion protocols, and many other people have been improving them alongside with us, improving expansion factor, optimizing it for different tissue types and sample types. And a couple of things have become very clear. Number one, it allows ordinary microscopes to become nanoimaging devices. And there have been hundreds and hundreds of published studies, either in preprint form or in peer-reviewed form, where people have used that aspect to do nanoimaging with equipment they already have access to. Number two, in recent years, a second benefit is becoming clear. By decrowding things for labeling, we can improve the labeling. And sometimes we convert invisible proteins or even invisible cells into visualizable form. Now, thirdly, looking forward, what's next? Well, one question is, how big can you expand, or how good can the resolution be? So when we expand twice in succession, the 20-fold expansion I mentioned earlier, a 300 nanometer resolution lens would now have a resolution of around 300 divided by 20, about 15 nanometers or so. Can we do better? Well, we and others have been trying to improve the polymer. For example, Ray, Gao, and Ju, and they were in my group, we worked on tetrahedral hydrogels that could expand specimens more evenly than the classical free radical hydrogels. So these have a diamond lattice shape, like you can see in the cartoon on the right. And we expanded viruses. We worked with Rachel Neve's group at Harvard to expand herpes simplex viruses because they have beautiful round shapes, and we thought, let's use them as sort of a natural scale bar. The take-home message is in panel G. The classical gel, as I alluded to, would introduce about 15 nanometers of error, but the new tetrahedral gel introduced around 9 nanometers of error. And that was borne out by preserving the shape of the viruses better. Recently, another European team led by Silvio Rizzoli with many collaborators, including some collaboration from our group, used a different hydrogel, this X10 hydrogel, which uses a different composition. It involves a monomer called DMAA, which is a self-cross-linking monomer. They also have been exploring improved resolution. They've been expanding tenfold and then using SRF, SRRF super resolution, which is a kind of super resolution microscope that can be done on confocal microscopes, and came up with some really interesting results. In fact, they are now able to expand individual proteins. So these are examples in panel E, where this is the cryo-EM ground truth of the GABA receptor, an important protein in brain science. In the right, where my mouse is pointing, they expanded these proteins and then imaged them on a confocal microscope. Now, how do they do that, right? You can't just pull a protein apart, right? It might stretch or warp. What they did was they anchored the protein to the polymer, chopped the protein into pieces, pulled the pieces apart, and then labeled them with a non-specific dye. Anyway, they were able to see shapes that looked a lot like the proteins that they're expanding. Panels F and G are another example of a ground truth in F and the expansion in G, and then I and J is another example. One of my favorite images from their bioarchive preprint is where they took the GABA receptor cryo-EM ground truth and then overlaid the expansion image directly on it. So panel A and B are the cryo-EM of the GABA receptor. Panel C is an example of an expanded GABA receptor protein, and you can see the 5-fold symmetry. So this is some nanobodies on the upper left. The two red dots are fluorophores. In the middle of the slide, you can see the expanded GABA receptor and the 5-fold symmetry, and then the overlay in the lower right shows a great registration. So this is very exciting, the idea that this might be getting down to nearly atomic scale in the years to come. Meanwhile, JB Chang, the same JB that I mentioned earlier, who used to be a postdoc in my group, has a bioarchive preprint on expanding the entire body, what he calls whole body expansion microscopy. And they've showed that they can take the entire embryonic mouse, soften cartilage and bone, and expand the whole body. So it's interesting to see that expansion might be the first technique that goes all the way down to nearly atomic precision, but all the way to entire mammalian bodies. Another direction is applying to different kinds of sample. So in Yongxin Zhao and Octavia Bekur worked with me, Astrid Wines, Andy Beck, and a whole bunch of pathologists at Harvard Medical School, for example. We showed that we could develop a version of expansion that worked on a whole variety of human tissues, not just brain. We showed it with, in the end, about a dozen different human tissue types. So on the left are normal, and on the right are cancer-containing specimens from prostate, lung, breast, pancreas. The color code is shown in the upper right. So again, we use fluorescent dyes to decorate different kinds of medically interesting molecule. And the take-home message is that in all cases, this protocol, which was designed to work on heavily fixed archival human biopsies and human tissues, was able to soften and expand them. So there's many purposes that this could serve in medicine. In nephrotic kidney disease, there are very small processes, podocytes in the kidney that can be ablated. Usually electron microscopy is used to diagnose these diseases, but with expansion, a regular light microscope can be used to diagnose them just as well as with the electron microscope in a small scale study that our group convened. Another example is early in a disease, it can be hard to detect the disease. In fact, that's almost the definition of early, right? The changes are small. We wondered, could we take those small changes and just make them physically bigger? So working with Stu Schnitt, a pathologist at Harvard Medical School, and others, we decided to look at early stage breast cancer. Stu and others had written a commentary piece that early in breast cancer, doctors can disagree about the diagnosis up to 50% of the time, half of the time. So we expanded specimens, and then Andy Beck did machine learning classification of the pre-expansion images versus the post-expansion images, using the votes of a team of pathologists to classify the specimens as normal, UDH, usual ductal hyperplasia, ADH, abnormal ductal hyperplasia, DCIS, ductal carcinoma in situ, different stages of cancer or normal states that are often assessed in human cancer biopsies. The group's votes were used to train the machine learning classifier, and then we did a cross-validation shown at the bottom. And the take-home message so far is that the post-expansion data resulted in much more consistent training of the machine learning algorithm than with the pre-expansion data. So our hope is that expansion might be able to potentially help take some of these early stage disease indications and make them large enough to be easily seen through classical pathology strategies. So to summarize, expansion is spreading very quickly. And I think part of the reason is that anybody can optimize it for their purposes. People are taking it down to nearly atomic scale. People are taking it to nearly or even the entire body. It's being applied to different kinds of tissues, different species. The list goes on and on and on. So this slide isn't really meant to be read, but just it shows a snapshot of the few of the many papers, experimental papers and preprints that have come out doing expansion. It's being applied to look at motor proteins, to look at the nucleus, to look at cell division, to look at the microbiome, to look at plants, and the list goes on and on and on. We've published the protocols for the most popular methods. So there's step-by-step protocols. It's a very easy to perform procedure and very inexpensive as well. And the process is very fast. There are forms out there that can be done in a few hours, even if you have a thin sample. Yongshin Zhao, who now is a professor at Carnegie Mellon, with some help from our group, published a sort of a rapid expansion pathology protocol that can be down to 4 hours in time. But even the classical procedures can be done in a day and a half or so. Pre-COVID, we hosted many visitors to come visit us and see the expansion protocols, but during summer 2020, of course, we couldn't do that. So during that summer, we published a step-by-step photographic tutorial on how to do expansion, including really simple rudimentary steps like how do you use a pair of forceps, or how can you use a paintbrush to pick up a specimen. And so even though during that time it was hard to host visitors and we were not able to host hands-on workshops, we were able to keep expansion spreading by having people look at the photographic tutorials. So I think I acknowledge along the way all the people who led those projects, but we've had many collaborators in our group and around the world for this and other projects that I didn't have time to talk about today. It's a very collaborative project, bringing together people from all over science, right? Microscopy, polymer physics, cancer biology. The list goes on and on and on. And so just want to acknowledge the great help from people all over the world on these projects. We have a website, expansionmicroscopy.org, where you can find the protocols and so forth, so please visit. And in addition to being the professor of the group, I'm also the chief tech support person, so just email me. It's just my name, Ed Boyden at MIT.edu. And if I don't know the answer to your question, we'll try to find it from one of our group members. And with that, I can take questions.
Thank you so much. That was really great. I mean, it's always great as a newcomer in the field to see how these techniques kind of develop and how it can just be applied to so many different things and just be made accessible to everybody. We did have a few questions in the chat. I can read them out, or I guess just one from George Campbell. He's asking, is the gel pore size known for compact and expanded gels? Is iterative expansion or a large expansion gel recipe, such as Trexem, needed for antibody entry into the gel?
Good question. So the pore size has been measured for these kinds of polymers through small angle neutron scattering and small angle X-ray scattering. And those studies go back many decades. That's how we know that the pore size is probably around 1 to 2 nanometers on average. We administer the antibodies before we form the polymer, when there is no gel, or we can administer them after expansion, and they can be brought in after 1 round of expansion or after 2 rounds of expansion. Yeah, so both work. You don't need iterative expansion to get antibody entry into the gel. The pathology brain cancer study that I mentioned was with 1 round of expansion followed by post-expansion staining. Yeah, great question.
All right.
Let's see, I can go ahead.
Yeah, do you want me to, I'll read out the question just in case people watching the recording don't have access to them. But so Claire asked, how do you handle image data management? What software do you use for your 3D rendering and videos?
Yeah, so much of what we do with imaging is using off-the-shelf software. We use ImageJ a lot, which is a free software that anybody can download. We have played also with other fee-based image software, and that also works well, and can sometimes offer nice specialized opportunities. We also write our own code. When we publish a paper, we always include all the code for distribution if we need a very specialized thing done. Yeah, for the 3D renderings that I showed, the animations were done by an animation company because it's more for like pedagogical purposes rather than like for scientific purposes. And then the fly through the fly brain was done by a visiting scientist, Yusuke Bando, who's from Kyosha, formerly Toshiba Memory, an expert on parallel computing. Yeah, so there's nothing really too specialized about it, but we work with our collaborators on whatever they want to do, I guess.
All right, and then Chris Chan asks, it's always difficult to see proteins of low abundance. Does the post-expansion staining consume lots of antibodies?
Well, if you have 100 proteins crowded together and can only label the outer 10, and now you're going to expand them and now you can label all 100, I guess you would be consuming more antibodies. But I guess the question is it worth it? If you don't really care about seeing everything, you could do pre-expansion labeling. And if you think about the roughly 450 or so experimental papers and preprints, the vast majority of them do pre-expansion staining. On the other hand, in our group, because if we do post-expansion staining, we often see things that are radically different than what we expected. We're doing a lot of post-expansion staining in-house just because it's showing us really surprising things. And once you know where they are, maybe you don't have to do it all the time, but a little bit of post-expansion staining can go a long way, is what we're finding.
Okay. And next question is from Brenda Jarvis. She asked, do you have a database of antibodies that work or don't work post-expansion?
Yeah, great question. I mean, the number of antibodies out there is nearly infinite, so we don't have enough money to buy them all and test them. Certainly the papers, we always disclose the antibody part numbers, and I would say ballpark, probably around 80% of the ones we try will work as well or far better than the original. By far better, I mean seeing things that we never thought we would see, like as I showed, the calcium channels or the amyloid plaques or the brain cancer specimens. We saw the brain tumor cells better post-expansion than with pre. Not all the antibodies work well, though. So if an antibody depends upon something very subtle about the structure, we do denature the proteins to expand them. So it's a little bit like an in situ Western blot, so to speak. My gut feeling is that if an antibody does work well in a Western blot, maybe it works better with post-expansion staining because we are treating the sample with heat and detergent before we expand the proteins apart from each other. But it'd be great if the community wants to make such a database. I mean, we can't afford to test all these antibodies for everybody. I know, but if the community wants to do that as a whole, again, with over 450 experimental papers and preprints, it's certainly something the community could do if it wanted.
Yeah, I think that would be something maybe our group can start tackling then if you don't have that available yet. So next question comes from Matthew Caffron. He asks, do molecular antenna dyes like BB421 and BB480 work for pre-expansion labeling?
I have not heard of these dyes before. I will try to learn more about them. If you can email me and send me a link to what they are, I'd be happy to look at the structure, and then we could try to see whether they would be hurt by the polymerization. I mean, so basically, I think what we found in the 2016 Nature Biotech paper was that you can add dyes to a specimen, and when you do the free radical polymerization, some dyes, like cyanine dyes, do get injured by the polymerization process. So if you know the structure, or we can do a check, we can certainly try to find out. But I don't, off the top of my head, know anything about these dyes.
Right, and the next question comes from Javier. For protein condensates, such as PSDs and synapses, and after expansion labeling, how do you balance accessibility of antibodies with loss of proteins due to protease treatment?
So to be very clear, when we do post-expansion staining, we don't use proteases. We take the specimen, anchor, polymerize, and then when we get ready for the softening step, rather than proteases for softening, we use heat and detergent to denature the proteins. We then pull the unraveled proteins away from each other. So for post-expansion staining, the epitopes are preserved because we don't use proteases. Now, more recently, we have started trying to use proteases that are kind of gentle proteases. Instead of obliterating a protein by chopping it into tiny pieces, they'll chop a protein into a handful of fragments. And that will then allow for the fragments to, for the most part, to still be antibody bindable. Why would you want to do that? Well, heat and detergent, might be great for certain purposes, like they really soften the specimen beautifully. But for example, if you're trying to study nucleic acids like RNA, doing aggressive heat treatment might be bad for the RNA.
Okay, and the next question comes from Elizabeth. She asks, how best could one retain and visualize lipid content?
Yeah, so there's several preprints and peer-reviewed papers now looking at lipids, although I think we'd all agree that's kind of early days. We have a bioarchive preprint on what we call membrane expansion microscopy. Recently, Yangshan and Zhao's group published a Nature Biotech paper on what they call magnify. And then there's several other papers where people are doing lipid stains with different kinds of expansion protocols. So many of the protocols will apply a lipid stain and then anchor the stain to the polymer and then expand. More recently, I think some of the more recent papers are trying to pull lipids apart and then label them, but none of those papers have been super quantitative about the amount of lipid retained, ours included. So I think by staining before expansion and then pulling apart the stains, I think that could be very quantitative, and we have a bioarchive preprint on a method where we do that. For pulling lipids apart and then staining them, that's very exciting, but I think there's a lot of quantitative validation that we and others still could do in the future.
The next question comes from Claire again. She asked, has anyone looked at cell matrix adhesion, and will they expand if they're adhered to glass cover slips?
Yeah, so many people have been looking at extracellular matrix. There's many peer-reviewed papers and bioarchive preprints looking at the glycocalyx or looking at perineuronal nets and so forth. So that works. Many groups are doing that kind of thing. As far as cells adhering to glass cover slips, so during the softening step, you take the cell, you apply the anchor, you form the polymer, and then you soften. During the softening step, things start to loosen up, and then very often the sample will pop off the glass. If not, you can try to take it off the glass with a razor blade. But in our experience, we would love to continue to use glass that doesn't adhere too strongly, and then for things to pop off the glass by itself.
All right, and the next question comes from Javier again. What is your experience with nanobodies?
Yeah, so our group does not have a lot of experience with nanobodies. I mean, kind of one of our ways of thinking of the problem is always to build tools that fit in with existing workflows, so conventional microscopes, conventional antibodies, and so forth. But others are doing nanobody work with expansion microscopy. I think there are the bioarchive preprint I mentioned earlier that was even expanding down to protein resolution from Rizzoli, the 1 nanometer expansion paper. They are using nanobodies, but it's certainly not my area of expertise. But there are other groups seeing success with nanobodies.
And then the last question so far comes from Nicolaus, and he asks, which would be your optimal solution for working distance problems with high magnification lenses and expanded tissues?
I guess the question is, what is the working distance problem that you're having? I mean, what we do is to try to buy lenses that have longer working distances, or to use specimens that are thin enough to fit in with the working distance. But if there's like a specific working distance problem that is sort of non-obvious, happy to to learn more about what you're struggling with. But if it's just a question of how do you deal with small working distances, I mean, that's a fundamental limit, right? So there, I think there's no secret sauce. Either we get a long working distance lens, but the good news is, people are making longer and longer working distance lens, or we can use thinner samples, but there's no secret sauce there.
Right, and then Claire also asked, so how do you store, backup, and share all the image data?
Yeah, good question. So there's two ways to think about it. One is sort of what are people actually doing? So if we look at like the roughly 450 experimental papers in preprints to expansion, a lot of people are not imaging very large volumes. Turns out a little bit of nano imaging can go a long way. And so very often a paper will have like, figure 1 will be conventional microscopy. Figure 2, we use expansion to study this little thing about it. And then figure 3 will be something else. Like, okay, we got our nano imaging out of the way. Now we're doing CRISPR or perturbing or doing something else. And so I think it kind of depends on the scientific question, right? Maybe you don't need a lot of image data. Now, as I alluded to, though, we are doing some experiments where we are trying to image entire brains and so forth, right? And those can be, I think, for that paper I mentioned where we worked with Eric Betzay to image the entire fruit fly brain and the entire thickness of the mouse cortex and so forth. I think we ended up with about a petabyte of data for that study, just one paper, right? And there, I don't know if we have any wisdom beyond what everybody else already has, right? There's cloud computing, there's servers, but I don't know if we have anything super insightful to add beyond what everybody already knows. That said, I do think there's a growing excitement as imaging does start to go into the petascale world, where many people are working on better stitching software, better distributed computing software, and the excitement about machine learning and AI and image analysis, is also pouring more and more GPU equipped computation into everyday biology. So yeah, this is probably a question that others could answer better than I could, but I'm very excited about all the trends that are benefiting imaging thanks to the pouring of talent and resources into the computational angle of imaging.
Right, and then the next question comes from George, and he asks, what is the preferred method to secure the hydrogel to a cover slip? Polylysine?
In-house, I think polylysine probably is one of our favorite methods. We have at times used like agarose because you can reverse, you can kind of get it out of the agarose, or super glue, which is very strong but finicky. But I think polylysine is kind of the often the first thing we try. But if you're struggling with some aspect of this, email me and I can run it by different people in the group, because even within our group, there are different styles of doing it depending upon what people want to do. So if somebody's going to do, we have a project now they're working on where we do many rounds of staining and imaging. And so there's sort of harsh treatments where we, kind of loosen up the antibodies for restaining. So there's sort of different kinds of constraints to think about. And so happy to hear what you're finding if you're struggling with something.
The next question comes from Izzy. She asked, for the approximate 4-hour histopathology protocols you mentioned, which steps in the protocols allow that speed gain?
Yeah, I think the basic idea was to speed up every step by taking advantage of the very thin nature of pathology specimens. I'll put a link in the chat window, but this is Yongshin Zhao, who's a former postdoc for our group, is now professor at Carnegie Mellon, is the last author of this. And so much of the key intellectual contributions did emerge from his group, I should point out. But yeah, if you're using 5-micron thick sections, you can kind of speed up the vast majority of the steps, it looks like.
All right, and I personally have a question. So for when you kind of image with like the really big brain slices, how do you kind of go back post image, like pre and post expansion, to kind of find that same area? Like I've been struggling that myself personally, so.
Yeah, so when we make the gel, we often trim it to have some kind of asymmetric shape. We often then take an image of the gel with a low magnification lens. Finally, we found a good use for our 4x and 10x lenses that we often didn't use for high resolution imaging, but we would take a low mag image. If there are fiducial markers in the gel itself, in the brain, we have the ventricles, we have different regions that we can use as landmarks. And this sort of just old fashioned getting to know the biology of it. So for the brain, where there's very stereotyped anatomy from mouse to mouse, that's been very helpful. But yeah, if it's something that you're kind of trying, the trimming of the gel, especially with an asymmetric shape, and you're struggling with landmarks and so forth, drop me a line. There are other groups trying other methods, but which we have not tried. But the beauty of this is that there's a whole community that's working on specialized versions for different kinds of problem. But in our hands, for the brain anyway, yeah, trimming the gel, knowing the landmarks, and using low mag lenses to get pre and post expansion images does the trick pretty much all the time.
All right, and then one more question from Brenda. Have you used detergents other than SDS for the softening step?
Good question. I guess you're playing around lots of stuff. Yeah, Triton and so forth, and also adding other non-detergent but softening things as well. So we have a preprint in bioRxiv. This is the expansion pathology decrowding, where we also add beta-recaptid ethanol to cleave protein-protein disulfide bridges to soften even more. So yeah, we are trying to use more and more softening agents for extreme cases. But I would say probably for many of our protocols, yeah, SDS, and I guess we are starting to play a bit with others, but has been the go-to.
Right, and I guess we have one more question that just popped in. If you don't mind staying a minute or two, do you think DNA can expand in a different way if it's not denatured or digested specifically? Great
question. I would love to see a very careful validation of expansion of the genome. So of course the genome is itself a polymer, right? It's what, a couple meters in length while it wound up into a cell. So it's a good question. If you expand it large enough, would you tear it or warp it in some way? Several groups have done 4-fold expansion and not seen any distortion with 4-fold expansion, but with 20-fold or more, then maybe it needs to be validated at that level of resolution. And I off the top of my head, I can't remember any example that's gone through peer review anyway, where somebody has expanded the genome 20-fold and done a careful pre versus post expansion validation. So, one idea that I've been trying to shop around is, take the genome, anchor it to the polymer, fragment it into pieces, and then expand the fragments, right? Which is, if you look at Hi-C or many other, or ChIP-seq, a lot of different genome mapping strategies that do depend on often some kind of nanoscale information. They do a controlled fragmentation of the genome, right? And that's a widely accepted thing to do. So if we could do that in the context of expansion, that could be great. And so that's not my core area of expertise, but I do think that if people want to expand the genome 20-fold or more, it could be very helpful to validate it very carefully the way that I described.
All right, perfect. I think that's the last of it. So I guess you mentioned if we have any other questions, we can contact you by email.
Absolutely.
And, perfect. Thank you so much for the great talk and for answering all of our questions. That was really helpful, I think, for all of us and for the community. Yeah, I think if anybody has anything else, if not, we'll let you go.
Great. Stay in touch.
Thank you.
All right. Have a great day.
You too.
Thanks.
Thank you.